I was just talking about someone earlier today. I had my undergraduate degree was just a pure biology degree, and everyone here is required to take physics, right? Then we had to also take the 2 intro chemistries, two organic chemistries, and then PChem, so physical chemistry. And then someone was telling me, a chemistry major was telling me, if you're just a pure chemistry major, you don't have to even take biology. You can just ignore it, which is interesting. So I think there's value, even if you're not specifically interested in genetics or the field, to be able to apply ideas from other fields to your field will make you stronger. Gives you more intellectual space and intellectual freedom. So I guess the main thing here is don't be afraid to take a class that's way out of your comfort zone. Because if you go about it right, I think you can apply it back. So, all right. So last time we left off just going over kind of what assay you use to figure out the cell cycle in Saccharomyces cerevisiae, what the readout. So we all kind of came to the consensus that we would mutate a whole bunch of yeast, right? Random mutation. We would select for a temperature-sensitive allele. So why are we selecting for a temperature-sensitive allele or temperature-sensitive mutant? What's the purpose of that? It's conditional, right? You can have, under some conditions it's going to be alive, in some conditions it's going to be lethal, right? You need to be able to propagate what you're going to use to study. So conditional, in this case, temperature sensitive. There's other ways to do this, but this is the simplest way to select one. So just drawing back on your standard biology knowledge, I'll take care of this. If you're wanting to make a temperature-sensitive mutation, what type of mutation is that? What type of mutation do you think is occurring in the DNA level and how is it being expressed at the protein level? Does that question make sense to you guys? So what type of mutation do you think would give you a TS allele of a gene? A nonsense mutation? You guys remember what nonsense mutations are? Right, nonsense mutation is the introduction of a start codon or a stop codon, right? So you think that would give you a TS allele of a gene? Probably not. That's going to give you a non-functional gene, right? So what types of mutations do you think could lead to a TS allele of a gene?
Point mutation.
Point mutation. So missense, right? So you're changing the amino acid. You're changing the amino acid in an essential part of the structure, right? Protein that's doing the work, the structure is changed in such a way that at high temperatures, if you're selecting for a TS mutant, it no longer effectively can carry out its job. Does that make sense? So if you're thinking about what type of mutation you're selecting for, it's a pretty specific type of mutation. You put some pretty intense limitations on your mutation, right? It has to be able to work in one condition and not work in the other, right? And I'll tell you, it's an open and honesty, that you work with different TS alleles of different genes, they're not all equivalent, right? Which should be hopefully fairly obvious, right? That some are going to be really tight, meaning that there's no phenotype at the permissive temperature whatsoever that you can determine, and some are going to be where there's a phenotype even at the permissive temperature, right? Or they don't completely die at the non-permissive temperature. We refer to that as leakiness, right? There's some, it's not exact on-off. All right. So, let's close out last time talking about the 3 individuals who got the Nobel Prize for the cell cycle. In 2001, the contributions, Lee Hartwell's contribution was screening yeast mutants for cell cycle control genes. Paul Nurse was important for doing that in fission yeast. And Tim Hunt was working with mitosis promoting factor in sea urchin eggs. Okay, so Tim Hunt was more of a classical biochemist. So we'll probably mostly focus on Lee Hartwell and Paul Nurse's contribution. So currently this gentleman here is retired. This guy is the director of the Royal Academy. So he just recently, you guys might have seen, he was in the popular press. You guys saw the GMOs, right? And he said, I thought that the British people were more sophisticated in their understanding of science until I read that 60% of British people don't want DNA in their food. Right? So hopefully you guys all realize that all of your food has DNA. Right? If it was living, it has DNA. So, alright. So just to give you guys a reminder, the yeast Saccharomyces through the cell cycle, you can tell where it's at by looking at the budding pattern. And you can grow as a haploid or diploid, which is going to be important for complementation. So right now I'm just going to take you guys through the experiments they did and why they were important. So you guys can visualize that. I'm going to go through this one fairly fast, but basically multiple events in the cell cycle. And you can tell by the amount of DNA, right? You can tell whether or not you've gone through synthesis or not, and then the budding pattern of the yeast where they're at. So using temperature sensitive mutants, they could halt the cell cycle during a mitotic division. Okay? So the reason it's a mitotic division is what? If you take a haploid through meiosis, what's going to happen? You can't take a haploid through meiosis, right? Okay. So remember they're working in haploids so they can see the mutations. Does that make sense to everyone? Alright. So you have a series where you have conditional mutants, right? You've mutated the proteins. Lower temperature allows for the function, a higher temperature reduces the function. So this is a classic TS mutation. There's also, it's called a cold sensitive mutation, or you can have mutations that are only responsive to other environmental factors. Okay? Also, some people actually made designer mutations, right? So for instance, if you were interested in a gene's function during development, say in a mouse or in a mammalian system, you can basically use a Cre-lock system, where Cre is basically a recombinase, so you can make conditional mutants. So when you express the recombinase, Cre, it cuts the gene out. So if you want to see, does that gene have a function in like liver development, right? You could put the Cre, which is the enzyme that's going to recognize the lock sites, which are flanking your gene of interest, under the control of a promoter that's only going to happen in the liver, right? So the whole rest of the mouse has this gene, has this protein, but then in the liver it turns on that Cre recombinase and gets rid of it. So you can see why this is a really powerful tool, especially when you start getting multicellular organisms, right? Another way to conditionally cause just those to have that gene excised. Does that make sense? So you can actually put a conditional mutant under an inducible promoter. So what do I mean when I say inducible promoter? We're going a little bit beyond here. Let's take a step back. Promoters. What do we think of when I say a promoter in a biological context?
A consensus sequence that tells you where to start.
Consensus sequence that tells you where to start transcription, right? Okay. So just as you guys all realize that in your body, all your cells are not expressing the same genetic information, right? They all have the same DNA. They're running different programs, right? Does that make sense to everyone? And what's controlling that is whether or not you're doing transcription of those genes, right? And some of these promoters are tightly regulated, such that they only turn on transcription under conditions. So this same system applies to single cellular organisms. The classic one that's used in Saccharomyces cerevisiae is a system where you can take a galactose-inducible promoter. So galactose is just another carbon source, right, that yeast can utilize to grow. You can put that in front of your gene of interest. And then that gene's only expressed if you have galactose, right? So now, if you take galactose away, that gene goes away. Okay, there's some complications there, but it's another way of conditional. There's a lot of ways to make conditional mutants, but TSs are the classical way. Okay. So because the alleles are rarer than loss of functions, they often have to isolate these from organisms that enable rapid high throughput screening, right? So you put a lot of conditions on these mutations, right? When you think about it, you're like, I need you to be screwed up here, but functional here. It's not an easy one to pass, okay? A lot of times you would actually isolate the same mutation. All right, so at the molecular level, I think you guys have a pretty good feel for this, but I just want to make sure everyone's on the same page. What's happening to your protein, right? So something's changing that condition right? In this case, temperature is changing the structure of your protein. The easiest one to think about is that at the permissive temperature, I have perfectly folded protein that works well, right? And at the non-permissive temperature with higher kinetic energy, I've disrupted an important protein-tertiary interaction. It could be a tertiary interaction. It could also be an interaction with another protein, right? So not all proteins are going to function by themselves. So we're going to talk about that one a little bit. But let's just imagine that it screws up protein structure, tertiary interaction. More kinetic energy, higher temperature unfolds. Therefore, it's lethal, right? Does that make sense? Okay. So everyone's on board with that. Let's take it a little bit further and ask a question. All right, let's say your TS allele mutation that you isolate is such that it affects the interaction with another protein. So your protein of interest that you've got a mutation in, this mutation at the higher temperature doesn't allow it to interact with another protein that's required for its function, right? Now, how might you go about experimentally determining that, if that was your hypothesis? So this would be your classic suppressor mutation, or suppressor, how could you suppress the original phenotype, okay? So we're getting more into this, but I want you guys to think, what would happen, so you have a TS allele, right, that affects protein A's binding to protein B, okay? So just think, biologists, we love blobs, right? So A normally binds B, then you get some sort of function, right? And that function is essential for cell. However, if you have a TS allele of A, this no longer binds with the same affinity, such that now you're dead, right? That essential function doesn't carry out. Just intuitively, how do you think you could force this reaction if you reduce the reaction here? Stoichiometrically. What would happen if you overexpress B? Right? So you can find a suppressor in B that if you overexpress B or make a complementary mutation in B that can suppress the mutation in A. You can see they're functioning together. It's essential function. Does that make sense? Right? So just things to think about. All right, so if you can't get a TS allele, you have to think about other ways. And the classic way of making mutations and determining whether or not they were lethal or not, if you should try to go for a TS, was making the mutation in a diploid, right? So you make the mutation in a diploid, it's covered by the wild type copy. All right. So Oliver discovered yeast. We've already gone through this, so I'll skip through. All right. Why was it necessary to use TS mutants to study the cell cycle? Is it completely necessary? You need a conditional mutant, right? You need a TS mutant, a conditional mutant for any process that's essential for cell viability. Does that make sense? So there can be something that affects the cell cycle that's not absolutely required for viability. This is a pretty essential cellular process to go from one cell to two cells. So most of them are TS mutants. All right. All right, so yeast. So what's the advantage of studying yeast? We've kind of gone over this, but let's just grab a couple. Why spend so much time on this? From last time to this time, why yeast? Yeah.
The cell cycle is relatively fast.
It's fast, 2 hours, right? 90 minutes basically to go from one cell to two if you're completely happy in yeast. What else?
1 N.
1 N, you can work in haploid or diploid, right? It's easy to go in between the two. What else? You guys are all engineers. This is a really important one.
It's inexpensive.
It's cheap. That's important, right? It's cheap. If you want to make something, you're going to make lots of it. You're going to make lots of money. You don't want to put a lot of money into what you're going to make it with, right? So it's cheap. It's easy to work with. And anything else? There's one other big one that's out there.
Non-pathogenic.
It's non-pathogenic, which is nice. It's always a good thing, right? Anything else?
It's a model organism.
It's eukaryote, right? If it wasn't a eukaryote, we probably wouldn't care at the level we do care, okay? So that's important. So how did they go about doing this? I think everyone has a pretty good idea of what happened. So you can propagate one in organisms, right? So it's that you know you have a mutation. You can plate those guys at 22 degrees, right? So you would actually, if you're thinking about if you're mutating them, you would always mutate them under 22 degrees, right? Because if not, you're going to lose them out of your population. Does that make sense? So when you're growing them and propagating them, before you treat them with a mutagen, you're going to have them at 22 degrees the whole time. Because you want it to be non-lethal at 22. Okay? If you're growing them at 30 and you mutated them, or 30's where they're happy, right? You got your mutation, you just lost your mutation. You see what happened? So it's kind of one of those dumb moments in science. Have you guys had those in the lab yet? You're like, oh, you actually have to add polymerase to get PCR to work. All right, so then you allow the cells to grow. So here you're at 22, 22. And then what you can do is replicate plate. So what does a colony represent? This is a key genetic concept anytime you're working with colonies.
So
It's a progeny from one cell, right? So it's a genetically identical population of cells, okay? So if you're doing mutagenesis, right, all these mutants were completely random, so when you separated them by space, allowed you to do this. I mean, this was actually a huge accomplishment to come up with solid media to do microbiology. So Pasteur never had solid media. Do you guys know this? All right. So then you can replica plate. So basically in yeast, I can tell you that we use velvet. You guys probably all seen the, velvet paintings of Elvis or stuff like that. Or you guys are fancy, have velvet pants or something like that. But anyway, you can basically just transfer a little bit of each of these colonies onto new plates. Okay, it's called replica plating. It's a pretty common concept. And then you separate those plates into your two conditions, right? So you expect everything to grow, right, at 22 degrees. And then you're looking for mutants that don't grow at 36 degrees, right? So what's the classic genetic term for this type of approach? So everything's growing except what you don't want. So this would be a screen, right? You're screening for mutations that don't grow, right? So as geneticists, as you guys all, I don't know if you guys will go on to be geneticists, but if you can do it, design selections, right? What's the difference between a selection and a screen? Genetic selection versus a genetic screen. Yeah.
would the selection be wanting your mutant only to grow on something?
Right. So selection, you're selecting for what you want, right? So for instance, you could do a transformation, right? You guys, I don't know if you guys have done transformations. New studio biology course, we all do transformations, right? We have your plasmid expressing ampicillin, right? And then you plate, or your beta-lactamase, you plate on the ampicillin. You now selected for only things that got that plasmid because everything else is going to die, right? So everything you're looking at is what you want. In this case, the thing you want is not there, everything else is, right? So you have to sift through everything. So it's a major difference between a screen and a selection. So sometimes there's no way around screens, but I would encourage you to think in terms of selections and make your life easier. All right, so you can identify the gene, and then you can examine its cell morphology, right? So can you tell anything about where this is resting in the cell cycle from this plate? So the colony didn't appear. Yeah, you can't. You might be able to focus really intently on this one cell and get a focal point, right? Because it was one cell, right? Did it go through a few divisions or not? Okay? All right, so what would this look like in culture? So on your left, we have permissive temperature at 22 degrees Celsius. What you can see is yeast happily growing. This is an asynchronous culture of yeast. hopefully you guys can all visualize that there's some with really small buds, some large budded. You have the whole array of the cell cycle represented. Does that make sense? Now you raise this mutant to the restrictive temperature. What you guys know about the yeast cell cycle, where are these guys arresting? Would you call it a G1 arrest? What do you guys think? It's hard, like when you actually have to teach these courses, you have to become comfortable with silence. And it's pretty much there. They're large budded cells, right? So where's a large budded cell in the cell cycle? Yeah, it's right before division, right? So you don't know. You can look stain at the DNA, right? Do you have one mass of DNA or two masses? Right? It could be something that happened during S phase. It could also be something that's happening in cytokinesis, right? Cytokinesis is failing, or it could be a G2N, right? Just large budded cells, right? So it's towards the end of the cell cycle, right? Okay. There's a little trick in yeast that we'll kind of go through. So after you have those mutations, right, by examining cell morphology, you can take them, because they're all haploids, and you can start mating them together. Does that make sense? So you can start organizing your mutants. So if you did this mutation analysis, you'd probably end up with a couple hundred, hopefully, different mutations. And you start mating them all to each other, right? And the ones that come out and still have the phenotype, and that don't grow at 36, those would be in complementation groups, right? Same gene or same pathway, right? Does that make sense? You screwed up the same process. More likely the same gene, actually, right? The same pathway, you'd still have complementation, right? If it's the same gene and you still have the phenotype of not being able to grow at a high temperature, it doesn't mean it's the same mutation in the gene. No, right? So there's multiple ways to skin a cat, although you've really restricted yourself. So sometimes it's the same, sometimes it's not. How would you go about determining if you've isolated it, figuring out if your alleles, right, of these TS mutants are the same? So you guys notice I probably present things very experimentally. So I think everyone probably knows. So if you were able to isolate these two genes, that you know are mutants, right? So right now you just know you have two mutations in this pathway that arrest at a certain point in the cell cycle. Do you know which genes they are? No. So classic genetics, which thankfully we don't have to do so much anymore because molecular biology has helped quite a bit, they would have had markers on every chromosome, right? So oxytrophic markers. And they would have isolated, oh, that mutation occurs on chromosome 12. The reason they did that is by how it was being transmitted with a known marker, right? So we'll talk about how you would go about finding it now. So these mutations, just to reiterate, cell cycle, basically wherever the protein is going to function is where you're going to build up in the cell cycle, right? So here's the question. Could you just raise the temperature and look at them immediately? No, you have to wait for everything to collect back, right? You want to go at least one cell cycle. Does that make sense to everyone? Because depending on if your mutation is here, you're going to have yeast in all these different phases of the cell cycle. You want to give them time to collect at where the mutation is, right? Okay. All right. So then the question becomes, have the two strains obtained the same mutation in the same gene, or just two different mutations that arrest you in the cell cycle? How could you tell that? Kind of beating this dead to a dead horse. Uh-oh. This should be straightforward. So you have two mutations, right? You have two strains, right? You mutated them, grown them at 22, you throw them at 36, 37, you look at them, they both have a G2M arrest, right? Both arrest large button. How do you tell if it's two genes that affect that process, or if it's the same gene?
Complementation.
You made them together, right? That's all you do is you made them together. All right, so if you made them together, if it's the first case, what are you going to see? They're still going to arrest at the high non-permissive temperature, right? This is the second case. Put them together, they're going to be fine. Does that make sense to everyone? Okay. So that's one way of using complementation. And this organizes all your mutations into all these different groups. Okay? All right. So another way to think about this, these are two mutations that came out of Hartwell's screen. One's CDC28 and one is CDC7. So both of these I really like because they work on DNA replication, and their essential targets, I will tell you, are DNA replication. So a CDC28 mutant at the permissive temperature, you see all phases of the cell cycle. You raise it to the restrictive temperature, let a couple cycles pass, and all the cells arrest without buds. Okay? So a G1 arrest. Okay? And if you were to stain the DNA, there would be just one copy of DNA. Okay? So DNA replication didn't occur. All right, CDC7. So you guys all hopefully picked up what CDC stands for. Yeast geneticists are pretty famous for this. It's pretty easy. Drosophila geneticists are a little crazy. Cell division cycle. Right? Cell division cycle in my mutants. Also in yeast, anytime you see a lowercase in the genotype, like a lowercase, italicized gene, that means it's a mutation. Right? If it was a wild type, it would be uppercase. Okay? All right, so CDC7, the permissive temperature, you see all phases of the cell cycle. You raise the temperature to the non-permissive, the cell arrests with buds. Okay? So it arrests with buds. So then the question becomes, how can I order these guys in the cell cycle? Right? So we know the cell cycle, but you're not sure which acts before what, right? So it's pretty obvious, hopefully, to you guys. But if you mated them together and made a double mutant, right? The mutation that's going to show up in this system is going to be the one that you can see after, right? It's going to go all the way around to the second mutation, right? So it ordered CDC28 in front of CDC7. Does that make sense? So this is basically an epistasis that CDC28 functions before CDC7. Okay? All right, so it turns out that CDC28 encodes a really important cell cycle. It's actually a kinase. So do you guys remember cyclins? What are cyclins? So I'm kind of shooting in the dark here because I'm not sure what is covered in your textbook, but I guess probably all heard of cyclins. So cyclins are proteins that cycle throughout the cell cycle. Okay? We'll talk a little bit about it. But CDC28 functions at start. And in yeast, three things separate in the cell cycle. There's like, before at start, three different pathways are set in motion. One pathway is you replicate your DNA, right? The other pathway is, do I start making a bud, right? So in yeast, that's the equivalent of, do I start getting all the extra lipids and all the other stuff to form a new cell, right? So bud emergence. And the third is, do I duplicate my spindle pole body? Which the spindle pole body in yeast is the equivalent of the centrioles, okay? So this is where CDC28 functions, okay? Which is we're called cyclin-dependent kinase. So the kinase, it will actually, CDC28 functions with a series of cyclins in the cell cycle that directs the kinase function. Do you guys remember what kinases do? What do kinases do?
Phosphorylates.
Phosphorylates, right? Okay, cool. So if you look at the yeast cell cycle, it's going to look a little bit like a mess. We can work our way through it. These are all a lot of the CDC mutations that have a role in the cell cycle mapped onto the cell cycle. So just to orientate you guys, here's G1, right? S, DNA synthesis. G2, your mitotic phase of the cell cycle, and you'll repeat, okay? And then here's the things that I talked about. Initiation, DNA synthesis, nuclear division, okay? So you also have to break down your nucleus, right? Spindle duplication, so this is the spindle pole body duplication, and this is the button emergence, right? So actually making new stuff for a new cell, okay? So that's how the cell cycle breaks down. And then all these numbers here are a series of iterations that Hartwell did to organize them all in terms of when they function in the cell cycle, right? Does that make sense? So he was actually staining the DNA, and you can actually stain for spindle pole bodies also to know if these duplicated or not. And all this has been worked out. Okay, so now we have mutations, right? And we have mutations, and we know we have a pile of separate mutations, right? Do we know what gene any of these mutations are in? You know, we have no idea, right? This is geneticists. We like to get all kinds of cool stuff and not really know details, and then figure out the details once we get a big enough mess to work with. Right? Does that make sense? So right now you have a series of mutations that affect different aspects of the cell cycle. You don't know where these mutations are. All you know is that they're different and they're unique. Right? Does that make sense to everyone? All right. So this is just going over the complementation groups tell us nothing about the locus of the mutation. Right? What gene have we mutated? All we know is it's unique. Does that make sense? Such that two different mutations placed in the same complementation group must have the same mutation in the same gene. But that doesn't tell us which of the 6,000 yeast genes we've screwed up, right? All right. How can a researcher identify which gene a CDC mutation has occurred in? How do you get down to what gene have I screwed up? You guys are all working on something similar, I think, in the lab. So when you do normal complementation, right? When you go from haploid to diploid, you've complemented every gene, right? Is there a specific way that you can complement only one gene at a time? What's the starting material for your PCR?
Primers.
What?
Primers.
Primers, that's true, that's true. How about the template for your PCR? Let's go with that. Be more specific in my language. I hope I know it right. What's the template for your guys' PCR?
Cdna Library.
A Cdna Library, all right. So you guys know this, you just are. All right, so Cdna Library. What is a Cdna Library? So how about this guy not answer? We'll go over here. Cdna Library, what is it?
DNA.
It's DNA, all right. That's good, that's good. Where does it come from? What's the idea of a library? You know Dr. Oxford would probably see this video and be like, man, they don't know that? All right. So Cdna Library is when you reverse transcribe all the RNA of a given cell type, right? So you reverse transcribe it into DNA, and then you clone the DNA, right? So the advantage of doing that is you get rid of introns. Does that make sense? Right? So now you have a Cdna library, right? That represents all the RNA that's expressed in that cell. Okay? So you have genes on each of those plasmids, right? So what's the word library mean? What's the difference between a library and just one gene on one plasmid?
The whole genome.
It's everything that's being expressed, right? So this is at the RNA level. So this is a Cdna library. Now, what about genomic libraries? So you guys know what genomic libraries are. We'll go, so, how about you, Ben? Genomic library. So you guys are lucky I don't know your names, they're even worse.
When you have like all the DNA from a cell.
Okay, and how do you have it? So it's a good start. You want to help them out?
Would it, I'm not sure, would it also have introns?
Yeah, it's going to have introns. So genomic library is just the DNA. Okay, so it's different than a Cdna library, right, which comes from RNA. It's going to have all the DNA of a genome, right? But how is it going to occur? What does it physically look like? I mean, so you can think of mating a cell, right, a haploid cell to another haploid cell as a library, right? But it has everything, all the DNA coming over in a whole new set. But that doesn't help us out any. That doesn't tell us what gene we're complementing, right? We need to know a specific gene that we're complementing. So what does a genomic DNA library look like?
Is it condensed?
It's condensed, sort of. It's not quite the word I would choose, but anyone else want to help out? I'm going to open it up now.
Is it separated into different genes?
So, separated into pieces. Okay, so this is, let's take a really simple genome, right? We're only going to be however many base pairs. This is our DNA. My drawing's pretty bad, so let me know if you can't follow along, right? So this is your DNA. So let's imagine this is the DNA of a yeast. And normally, you have your mutant yeast over here. I'll draw a proper yeast. I can draw a bud. That's within my skills of drawing. Right? And you're going to mate it to this yeast. Right? You have a mutation somewhere here. Right? This is a S mutation. Then all this DNA is going to come in. It's going to now be, these are both 1N. Right? And this will be 2N. Right? After they get it on. Make sense? All right. So now you've isolated the DNA from this guy, which you know doesn't have the mutation, right? The problem is you don't want to just use all the DNA because it doesn't tell you anything about which gene did the complementation, right? So you isolate the DNA from a wild type yeast, right? You cut it up. What can you use to cut up DNA? Restriction enzymes. All right. Now we're talking, right? You use restriction enzymes. So let's make this easy that you have A, B, C, D, E. We'll just call those our genes on our fancy DNA, right? Now you have all the DNA in bite-sized pieces, right? Because you've digested it with the restriction enzyme. Now, how can you get this DNA into this cell? Could you just transform the piece of DNA into the cell? So that's one we're not going to talk about, because probably you could, but we're not going to go into that. That's not going to tell you because you're not going to be able to get it back. I'm not going to worry about that. What's a way that you can transform DNA into a cell? Plasmid, right? So take a plasmid, and you use complementary restriction enzymes, right? Complementary sticky-ins. And you ligate, right? So you guys remember where ligase comes from, right? This creates a new phosphodiester bond, right? So now if you make a library of this genomic DNA, right, you're going to have a situation like this. Right? With all these gene pieces, right? Represented in a plasmid library. Does that make sense what a plasmid library is now? Okay, now you're going to do a transformation, and transformation is really inefficient, right? So more than likely you're only going to get one of these plasmids transformed, right? So then you're going to have tons of these yeasts, each transformed with a different plasmid. right? Now, which one's going to grow at the restricted temperature?
One with a mutation.
The plasmid, so let's say this mutation was in B, in our simple example here, right? The plasmid that contains the genetic information for B, right, is now going to complement this haploid yeast because at that gene loci, it is now diploid, right? Okay, so you still don't know what was what in your library and all that good stuff, right? But now you've done a selection, right? Because the only one of these plasmids in your library that's going to grow, hopefully, this is under best case scenario, is going to be the one that complements this gene, right? Now you have this guy here with B on a plasmid, okay? So you get complementation. Now you need to isolate that back out, right? So it's just a plasmid rescue from yeast, right? Plasmid prep, and then you sequence B, and you know what that gene was that affected the cell cycle at that stage. Is everyone with me? All right, so let's go through it in something better than my drawing. All right. So put Cdna library into yeast cells that grow at the restricted temperature. So you can use a Cdna library or a genomic library. There's limitations on both. What's a limitation on a Cdna library? The Cdna library have equal representation of all your genes. No, it's biased, right? How's it biased?
It's only the ones that are expressed that are actually here.
It's the ones that are expressed, right? I can tell you express a lot more of like your housekeeping genes than you do enzyme genes, right? So this is a pretty essential cellular process that's highly regulated. So you're probably not expressing a lot of these cell cycle proteins at high levels, right? What about a genomic library? Are they equally represented in a genomic library? Right? And I drew our example really easy, that we had each gene cut perfectly with a restriction enzyme. Is that going to happen in real life? No, of course not. So you do what you call a partial digestion. It's not hitting every restriction site, so you always get overlap. So a real library would have a fragment that's here, a real fragment that's here, right? So usually you shoot for genomic library 8 to 12 kb. Okay? Does everyone understand what I'm saying when I say kb? Alright, cool. So we have your library on your plasmid. What all do you need on your plasmid? What do you need on a plasmid? It's all there. Let's talk about what each of these do. So LU2, what's LU2 on this plasmid? What's it sound like? A promoter? Maybe. It's not a promoter. So if you had a Cdna library, you would need a promoter, right? That is true. Here it's a way to select for the plasmid transformation in yeast. So what I'll tell you is that LU2 is essential for leucine biosynthesis. So if you grow in the absence of leucine, right, and your strain is mutated for LU2 gene, you're complementing that gene to select for the plasmid. Everyone see that? So the cool thing when you're doing essential genes is you really don't need that selection, because now the gene that's essential that you put in also complements and selects for the plasmid. You guys see that? Because everything else is dead, which is always good. All right, yeast ori. What the hell is that? Origin of replication, right? Your plasmid has to be able to replicate. Yeast DNA insert. Okay? So this is the insert of the yeast DNA. If you don't know what it is, it's red. Okay? All right, and then there's a couple other things here that are just tricks. There's an M persistence and an E. coli origin. Why might those be useful?
Well, if you wanted to express that particular DNA sequence in an E. coli bacteria.
Right. So this is what's called a classic shuttle vector. You're going in between yeast and E. coli. Okay? So this plasmid can be propagated in both yeast and E. coli. Does that make sense to everyone? So E. coli is kind of the workhorse. It's an easy place to make lots of genomic DNA, or plasmid DNA. All right, so each yeast cell picks up a different plasmid, thus a different wild type gene, so a different piece that can do complementation, right? Cells that pick up the wild type copy of the gene they lack will be able to grow at the restricted temperature, right? And then you can isolate that back out. So all that's going to grow here at this restricted temperature is the one that complements your original mutation, right? You can isolate that plasmid back out, and bada bing, bada boom, you have your gene that affects the cell cycle at that spot. Okay? All right, so I'm a little bit behind, but we'll get there. All right, so this is a list of Pombi and Cerevisiae, all the genes that were originally kind of found that are important for the cell cycle and found in these screens. So before this, why it won the Nobel Prize, people didn't understand how a cell went from one cell to two cells in the process. So this is probably, a lot of this work, late 70s, early 80s when it began. So I know my postdoc advisor actually worked for Hartwell. He worked on CDC7. So CDC7 was just a number that didn't know what it did. And that was 70s. All right, so now, what do we know about evolution? This is why we care to do it in yeast in the first place. The really cool thing is, you can take a cDNA library from humans, right? This is an essential cellular process. The more essential the cellular process is, the more highly conserved it is, for the most part, okay, in general. You can take a cDNA library from humans, and now you can identify the genes that carry out that function in humans because you'll get complementation. Everyone understand how cool that is? So now you've complemented with a human gene that's doing a similar function in human cells that can replace the function in yeast cells. Which is pretty amazing, right? So it allows you to start identifying things that control the cell cycle in humans. Because unlike yeast, we don't have a quick, easy way to do all this, right? It's getting better, it's getting better. It's pretty amazing when we think about it. All right, so key concepts, right? Cell cycle, cell division cycle, cyclin-dependent kinase, which is CDC28. Right, we didn't talk about too much about the cyclins. A TS mutant, right? Why do you need a conditional mutant? Complementation groups, okay? So all that good stuff. Provide 5 reasons why yeast were used in this study. I think we covered that earlier. You guys pretty confident in that. Make sure you guys understand that. Describe why temperature-sensitive mutants were used to study the cell cycle, right? You guys can come up with your knowledge of molecular biology and think of other ways you can do it today. There's multiple other ways. Given the results of complementation cross, define the number of complementation groups and the number of genes needed for cells to perform the activity. So we didn't really go into that too much, but your complementation groups, you can basically group them all into, you have this many mutants, right? Does that make sense? So you could start off with 100 mutants and end up with 10 complementation groups, right? And then you can figure out where they fit in that cell cycle. They have stasis. So we talked about that briefly. All right, so the last few minutes here, now you have the genes, right? Whoop-de-doo, right? I mean, it's progress, but now you know the genes, but you know nothing about them. So I want to have a class discussion, and I want you guys to brainstorm. Now that you know the gene that's important for the cell cycle, where would you go with it? What would you do? All right, protein, right? So obviously you hope the gene makes a protein. If not, it's a little bit more difficult. But you can express the gene, right? What's an important aspect in function from expression? So what could you figure out in expression? So if the gene was under its normal promoter, because you know what the gene is now, right? And you think it's important for, let's say, cytokinesis, right? When in the cell cycle would you expect to, if you isolated yeast cells, I'm going to tell you that you can synchronize yeast cells in the cell cycle. Turns out when they want to mate, they all stop at G1, right? So you can stop them all there. Where would you expect to find yeast cells, that protein expressed, if it was important for cytokinesis? The variant of the cell cycle, right? You wouldn't expect it to be expressed in G1 if it's being utilized and controls the cell cycle at cytokinesis, right? So you can get a temporal protein expression. So we're off to a good start, right? What else? I mean, we know nothing about what this gene's actually doing, right? How can you figure that out? Yeah?
You can see where it's actually being used in the cell.
What's the cellular localization of this guy, right? So we have the timing. When's it expressed? What's the cellular location, right? Yeast, cellular structures aren't awesome in yeast, but you can definitely tell if something's nuclear, right? Or not, right? Cytoplasmic, or is it in the nucleus, right? So timing that. What else can you do with this guy? What it's interacting with. So there's multiple ways to go with what it's interacting with. So geneticists, what we like to do, and I can tell you this for next on CDC7, you look for things that suppress the original phenotype. Right? So you have the original TS phenotype of, at the non-permissive temperature, this doesn't grow. Can I get another mutation that now suppresses that initial mutation? Right? And that's going to tell me genetically that those two are related somehow. Right? So I can tell you the guy who I did my postdoc with did that for CDC7. And what it all comes out, and this is years later, and we'll just focus on CDC7 and CDC28. They're both important for the initiation of DNA synthesis. So CDC7 actually turns out to be a kinase also, and its major role is to phosphorylate the MCMs. So his suppressor mutation that he was able to isolate was in the MCMs. Right? So then you have to figure out where your suppressor is. And then you have two genes, and you're like, okay, if this is a mutation, then I change this mutation and suppresses it. Right? So somehow they're genetically related. So you just keep doing those iterations. Right? The other approach, what happened really nice at the same time, just want to kind of finish on this idea, is that people knew about mitosis promoting factor, right? There was something you could take from mitotic cells, the cells that were in mitosis, put it into other cells and cause them to go into mitosis, right? What that turned out to be was MPF is what it was originally called. And what that turned out to be was cyclin-dependent kinase with cyclin B. Remember, this is CDC28. So concurrently, a biochemical approach had identified this gene for being important. Right? So now you have genetics and biochemistry coming together to understand cellular function. Does that make sense to everyone? So the key word now is you have to be able to do biochemistry, genetics, all the above to be successful. Okay? All right. I will see you guys probably, I don't know. Good luck.